noticed I went to Disney. That's good. I hope you enjoyed yourself. It's been a while since I've been to Disney. Like I said, I was going to do a photo book of all the pictures from Disney. Which park did you go to? Or did you see all of them? Magic Kingdom. Okay. Yeah, that's a good place to start. Being the scientist that I am, I always like the animal kingdom as well. But all the parks are nice. I haven't been there since they added a lot of the new attractions, but Disney is definitely one of those things that if you've never been, that should be on your bucket list. It's getting more expensive to go, but it's definitely worth going to see. It's really a great experience. All righty, let's see. We got almost everybody here, so we'll go ahead and get started. No, not everybody. I'll give it another minute. Anybody else do anything? Only one person shared what they did over the holidays. Anybody else want to be brave and share what you did? I did a lot of life science. Like with the place where I was staying, they just had access to all kinds of things. I fed some horses. One of them was really mean and wouldn't let the other horses get the food. I went to a birds of prey exhibit. So I got to see a horn owl up close. Well, I forget what the little one, a hoot owl, screech owl. The little baby one was a screech owl. Not, he's not a baby; he was full grown, but it's so tiny, about the size of your hand. And then they had a very large um, red-tailed hawk. I might put together a little slideshow because that was so interesting to see all of those animals and what they do. But that's just a science person in me. Went to another presentation where they talked about the wildlife in the area, especially the alligators. They tell you beware the alligators. And so I learned some interesting things about that. But those were the highlights of my trip, other than being able to walk the beach. And I saw dolphins swimming and jumping. So that was interesting for me. That's because I'm a life science kind of person and I like stuff like that. So that's not why I went, but it just turned out to be a plus to my trip. All righty, almost everybody's here. So we'll go ahead and get started. All right. So we are on that last leg of this very important course, biology, that you have to take the milestone exam for in April. So you want to make sure that you don't throw away anything that you had from last semester. Make sure that you keep all of those in a safe place. Uh, you can, I don't know if you created a separate folder for all of your work, but you should keep it because it's too much stuff to just send you a study guide. Later on, I'll send you the link to the Georgia website that has the milestone study guidebook so that you can use that. You're welcome, Keone. Um, so that'll be important, but we'll also take time starting in March. We'll start taking little breaks to go over old topics as we prepare for the exam in April. Okay. Like I said, most of you did very well on um, the test y'all took at the end of the year. The name of it?
Midterm.
No, not the midterm. With the one, the general test that y'all did, and the name of it is escaping me right now. But you'll have to do it again. So, and you'll see that same test again. Um, with the same questions, and hopefully there'll be more things on it that you'll know. Like for example, a lot of it I'm sure is going to be on the things that we're about to do now, which is cell division. So, oh, why can't I think of this name? Benchmark. There you go. I had to look it up. Benchmark test. So you will see that benchmark test again. Like I said, most of you did very well. Was not for a grade, but it was an indicator of how much information you already know and how much information you need to know in order to be successful on the exam in April. So that's the way you were to look at it. So again, you'll see in March, we'll take that test again, and hopefully you'll know some of the things that you kind of guessed that we had not covered yet. All right, so that being said, just let a couple of more people in. That was kind of just hanging out for a minute. All right, so be in Canvas. We're on the unit cell reproduction. So before we left, we talked about DNA and protein synthesis. So we looked at the structure of DNA, and now we're going to look at how that structure of DNA is replicated for cell replication. So we talked about how the DNA makes a copy of itself, but now we're going to take that a step further and look at how it is used as the blueprints for reproducing a whole new cell, and what our genes look like and how it's affected. So we're still going to get deeper into mutations, because that time I told you that there were two sets of mutations that we looked at. We only looked at one set, and now we're going to look at the other. All right, so if everybody logs into the overview is very important because it summarizes all of the key points and vocabulary. There's a lot of vocabulary with this. This is not an easy section. It's going to have a lot of diagrams, a lot of things you're going to have to label. And so it's going to be important that you stay focused, that you're not playing games when you should be paying attention, and that you are actually logged in and doing what you need to do. We want to make sure everybody does not fall behind. So again, please use class time for class work, so that you can stay on top of things, because the milestone will be here before you know it. And again, I want to remind you that the milestone is 20% of your grade. So I'm encouraging all of you to shoot for having an A or a B in the class, so that if you don't do so well on the milestone, it doesn't pull you down to failing the class. You fail the class, you got to do it all over again, because biology is a mandatory class. Okay? You need it to graduate, so you need to take it seriously. And some of y'all are not on camera. You need to be on camera. That's a school rule. So don't some of you logging in and you're just here to be hanging out for attendance. So please be on camera, on camera. All right. So again, this is a overview. So as you can see, there's several aspects of this. We are going to take each of these as a module. So there's a module on mitosis, there's one on meiosis, there's one on asexual and sexual reproduction, because there is a difference between those two types of reproduction. And then we're going to look at, why do we have cancer? Cancer is really just when cells reproduce out of control. Some little mechanism has clicked in and it is reproducing in areas where it shouldn't be reproducing, and it is reproducing out of control, which is why you get certain growths that appear, or certain cells that take over, that destroy the good cells by creating their own space. So we're going to look at all of those. So as you see here, we've got quite a list of vocabulary words. So we're going to spend a good time in trying to make sure that you understand each of those as they apply. So again, we're going to talk about different types of mutations than the ones that we talked about the last semester. And then all these other things will come into play. All right, so that being said, now that I think everybody is here, not everybody, let me see, one or two people, but for the most part, everybody has decided to start the year off right and attend class. Some of you are still not on camera, but you need to be. All right, so before we get into this, I'm going to show a short video on cell division in general. So like I said in the previous one, there are different types of cell division. The first one that we will attack, it'll be mitosis, but I'm going to show a short video on okay, I think I put it in your list already, so let's see if I can get to it from here. All righty, there it is.
Good morning and welcome to Biology. I'm Paul Suchecki and this is my classroom. It's great to see everybody's smiling face again today. Today's lesson is on cell reproduction. You know how living things grow. You're a living thing. You still growing? Probably. I stopped growing a long time ago. Trees grow, grass grows. Oh, I just remembered. I gotta cut my grass this weekend. One way living things grow is by cell enlargement. If their cells get bigger, the entire organism gets bigger. Another way is by cell division, where one cell divides into two. 2 cells become 4. 4 cells become 8. 8 cells become 16. 16 cells becomes 32. 32 cells become 64. I think you get the idea. The more cells you have, the bigger you are, right? Think about an elephant. Think about a mouse. Does an elephant have bigger cells, or does an elephant have more cells? Are a mouse's cells smaller, or does a mouse just have fewer cells? Cells come from pre-existing cells. In other words, cells make cells. Cells make cells by a process called cell division. One parent cell divides into 2 daughter cells. I don't know why they're called daughter cells. I think they should be called son cells, or at least...
We'll pause him for a minute because he talks very fast. All right, so one of the things that you have to remember, and we talked about, there's the cell theory. We talked about that last semester, but the most important thing about the cell theory is that cells come from pre-existing cells. They don't just pop up out of ooze. A cell has to make a cell. Okay, so that's important fact number 1.
Let's be gender neutral and call them sibling cells. Anyway, nobody invented cell division. Nature invented cell division, and it's been going on continuously since the beginning of living things. Cell division is how multicellular organisms grow. It's how unicellular organisms reproduce. In a eukaryotic cell, the DNA is located inside the nucleus, and it takes the shape of a double helix. Think of DNA like a long thread that stores information. Most of the time, the DNA is just lying around the nucleus in a loose, unraveled form called chromatin that's wrapped around special proteins called histone proteins. I call chromatin the working form of DNA. When the DNA is in the chromatin form, the cell can read it, follow its instructions, and make any protein that the cell needs. Just before a cell divides, but after replication, the DNA is supercoiled into little bundles called chromosomes. Chromosomes are bundles of supercoiled DNA. Each chromosome has 2 halves.
Okay, I'm going to stop right there. So again, the second important part about this lesson that we're going to do is the chromosomes. So before we were just talking about the DNA and the double helix. So now we're going to look at the fact that all of that DNA is super condensed in the nucleus, and then what it's going to do during this process is going to spread out so that we can actually see what's going on. But there are all kinds of chromosomes in your nucleus. Again, important part, all of this is happening in the nucleus of the cell.
Half is called a chromatid. The 2 chromatids are mirror images of each other. They're the product of DNA replication. They hold the exact same genetic information, or at least they should. The 2 sister chromatids are held together near the middle by a structure called a centromere. During cell division, the centromere breaks apart and the 2 sister chromatids are pulled into opposite daughter cells. Chromosomes always come in pairs, just like shoes, just like gloves, just like pants. Humans have 23 pairs of chromosomes for a total of 46. Pairs of chromosomes are homologous. That means they're similar, but they're not identical. Kind of like your shoes. What I mean is, let's say there's a gene on a chromosome right about here for eye color. On the homologous pair, there's also a gene for eye color in the exact same location, but it might not say the same eye color. One chromosome might say blue eyes, the other chromosome might say brown eyes. But the gene for eye color is in the same spot on both chromosomes. That's what I mean by chromosomes are similar, but they're not identical. You received one chromosome from each pair from your mom, and you received the other chromosome from each pair from your dad. So of the 46 chromosomes, 23 of them, one from each pair, came from your mom, and the other 23, one from each pair, came from your dad. And you're the genetic combination of both of your parents. It's kind of like taking a half a deck of cards from your mom and a half a deck of cards from your dad and shuffling them together, and then let's see what you get. That's you. Normal body cells are called diploid because they have the full number of chromosomes, that is, 46 in humans. We use the symbol 2N to represent a diploid cell. When I say normal body cells, I mean things like skin cells and liver cells and bone cells and muscle cells and, all of your body cells. Gamete cells are haploid, meaning that they only have half the number of chromosomes. Gametes are reproductive cells, sperm cells in males, egg cells in females. We use the symbol 1N to represent a haploid cell. I hope it makes sense that gamete cells are haploid. The sperm cell is haploid, 1N. That is, it has half the number of chromosomes, 23 in humans. The egg cell is also haploid, 1N. When a sperm cell and an egg cell come together to form a zygote, the zygote is now diploid. It has the full number of chromosomes, 2N. Before that zygote divides to form two cells, all of the chromosomes replicate, and then the zygote divides. That way, each daughter cell will have the full number of chromosomes. When cells divide, they go through a cycle called the cell cycle. During the cell cycle, there's an interphase between divisions. Some people call it a resting phase, but oh no, the cell's not resting during interphase. The cell's taking in nutrients, it's growing, it's synthesizing materials, it's replicating its DNA and getting ready to divide. I wouldn't call that resting. Interphase is the longest phase in the cell cycle, and it's divided into 3 subphases: G1, S, and G2. The G in G1 stands for growth. During the G1 phase, the cell's taking in water, amino acids, fatty acids, carbohydrates, and these materials are becoming part of the cell, which makes the cell grow. The second part of interphase is the S phase. The S in S phase stands for synthesis. During the S phase, a duplicate copy of the DNA is synthesized by a process called DNA replication. The purpose of DNA replication is to make sure that each daughter cell gets all of the genetic instructions. During the G2 part of interphase, the cell's making its final preparations and getting ready to divide. The division of the cytoplasm and all of the organelles is known as cytokinesis. There's usually enough of each organelle so that when cytokinesis occurs, each daughter cell gets the organelles it needs. Of course, there's just one small problem. There's only one nucleus. So before the cell can divide, the nucleus has to divide. The division of the nucleus is called mitosis. Mitosis occurs in 4 phases: prophase, metaphase, anaphase, and telophase. Just remember PMAT. Sorry, Matt. Remember, we define mitosis as the division of the nucleus. Interphase is not one of the phases of mitosis. Interphase is the in-between phase, when the cell's not dividing. So by definition, we can't include interphase as one of the phases of mitosis. I've seen this error before. It's not IPMAT, it's PMAT. The first phase of mitosis is called prophase. During prophase, the nucleolus and the nuclear membrane disappear into the cytoplasm. The unraveled chromatin condenses and supercoils into chromosomes. 2 structures called asters or centrosomes appear at one end of the cell. One of the asters migrates to the opposite side of the cell. The second phase of mitosis is called metaphase. During metaphase, the chromosomes line up down the middle of the cell. There's got to be a way that you could remember that during metaphase, the chromosomes are lined up in the middle. Metaphase, middle, metaphase, middle. I'm sure you'll think of something. Also during metaphase, spindle fibers grow from each aster and attach to the centromere of each chromosome. The third phase of mitosis is called anaphase. During anaphase, the chromosomes are pulled apart. You see, the spindle fibers at opposite ends of the cell start to pull and play tug-of-war on each chromosome. This breaks the centromere apart, and the sister chromatids are pulled to opposite sides of the cell. Anaphase, apart. Anaphase, apart. There's got to be a way that you can remember that during anaphase, the chromosomes are pulled apart. I'm sure you'll think of something. The last and final phase of mitosis is called telophase. During telophase, the cell membrane pinches down the middle and forms what's known as a cleavage furrow. The cytoplasm is beginning to divide. Cytokinesis occurs, all of the organelles in cytoplasm are divided up, and we end up with two identical cells. The two daughter cells are now half the size of the original parent cell. In plant cells, since there's a cell wall, a cell plate is formed between the newly developing daughter cells. At the end of telophase, there are two cells. Telophase, two cells. There's gotta... You'll think of something. So what comes after telophase? Interphase, of course. The chromosomes unravel and become chromatin again. The nuclear membrane reappears and surrounds the chromatin. The nucleolus also reappears, and the two daughter cells go right back into interphase. They spend most of their time in interphase, growing, synthesizing, and getting ready to divide again. Remember the cell cycle? Prokaryotic cells like bacteria don't have a nucleus, but they still divide. Since they don't have a nucleus, and mitosis is defined as the division of the nucleus, we can't really call prokaryotic division mitosis. When prokaryotic cells divide, it's called binary fission. Prokaryotic cells don't have pairs of chromosomes. Instead, they usually have a single circular chromosome. During binary fission, this circular chromosome replicates, and then the cell simply splits in two. Compared to mitosis, binary fission in prokaryotes is relatively simple. The circular chromosome begins to replicate at one point in both directions. When the two replication forks meet, replication is done. Cytokinesis occurs, and the cell divides in two. Easy peasy. Meiosis, or meiosis, is it meiosis or meiosis? Anyway, meiosis is a special kind of cell division. Meiosis is the production of gametes, reproductive cells. In males, sperm. In females, eggs. Sometimes we call meiosis reduction division, because during meiosis, the number of chromosomes is reduced by half. In meiosis, we start with a diploid or two-end cell. The nucleus divides twice, and we end up with 4 haploid or one-end cells. See, the number of chromosomes is reduced in half. Reduction division. Why do you suppose gametes are haploid? Gametes have half the number of chromosomes. They're haploid, so that when they fuse together to form a zygote, the zygote is diploid. It has the full number of chromosomes. This does two things. One, it promotes genetic variety. The new individual is a genetic combination of both parents. But secondly, by reducing the number of chromosomes in half during meiosis, it ensures that the same number of chromosomes is passed on from generation to generation. In humans, that would be 46, 23 pairs. In order to reduce the number of chromosomes in half, during meiosis, the nucleus divides twice. So there's actually a meiosis 1 and a meiosis 2. During meiosis 1, there's a prophase 1, a metaphase 1, an anaphase 1, and a telophase 1. But that's just the first division. During meiosis 2, there's a prophase 2, a metaphase 2, an anaphase 2, and a telophase 2. When it's all said and done, we end up with 4 haploid cells. One of the main purposes of sexual reproduction is to promote genetic variety, genetic diversity. This is achieved a couple different ways during meiosis. During metaphase 1, Instead of all the chromosomes lining up down the middle in a single file line, they line up next to their homologous pair. This is known as a tetrad. Then something interesting happens. Homologous chromosomes start trading little pieces of each other in a form of cellular genetic reshuffling that promotes genetic recombination and increases diversity. After crossing over, the homologous chromosomes are then sorted independently by a process known as independent assortment. In other words, the chromosomes are separated randomly, and this further increases genetic diversity. In males, meiosis occurs in the testes and produces millions of sperm cells every day starting in puberty. The production of sperm cells is known as spermatogenesis. In females, meiosis occurs in the ovaries, resulting in about 400 egg cells in a lifetime. These egg cells are formed in the individual's ovaries before they're even born, but they're not released until the individual reaches puberty, and they're usually released about one per month. The production of egg cells is known as oogenesis, or do you say oogenesis? Is it oogenesis or is it oogenesis? During oogenesis, 4 haploid cells are produced, but only one of them becomes an egg cell. The reproductive organ for most plants is a flower. Usually, flowers have both male and female parts. The male parts produce pollen. Each pollen grain contains male gametes. The female gametes, or the egg cells, are produced in the ovary of the flower. Pollination occurs when pollen is moved from the anther to the stigma. The pollen moves down the style of the flower and into the ovary where fertilization occurs. The zygotes develop into embryos. The embryo is hidden inside of a seed. Eventually, the seed falls off the plant, lands on the ground, and germinates into a new plant. So anyways, all living things grow, and one way that they grow is by cell division. You know, I asked you a question at the beginning of the lesson. Do elephants have bigger cells or more cells than mice? Well, it turns out that elephant cells and mice cells are about the same size. If you were to look at a little piece of liver under a microscope from an elephant and compare that to a piece of liver from a mouse, the cells would look the same. Elephants are bigger than mice because they have more cells, but their cells are about the same size. Anyways, that's about all I have for today, so I'm going to leave it there for now. Until next time, don't get lost and have a great day. See you later.
All right, so that was an overview of the unit. He basically went through your whole vocabulary list. And we're going to teach each of those things. Again, that's in your playlist, so you can go back at any time and look at that overview. He covered just about all of your vocabulary words in that this video, so that you have a visual on what cell reproduction is. So again, coming back to the overview, there's different types of cell division. So we're going to start with mitosis. And by the way, he said how you pronounce certain things. I say meiosis. I do not say meiosis. And I say oogenesis. I don't say oogenesis. So mitosis. And we're going to look at that to start with as our first basic. Most of the cells in your body are going to do mitosis. Okay. And again, this is why he said that the mouse and the elephant are different sizes, is because there's just more cells in an elephant than there is in a mouse. But a cell is a cell. So we are going to go back and look at cell function. That's something we did last semester. Everybody did different parts of the cell. So we are going to look at that again, just a little bit in review to know what the different things do in the cell. But mostly this semester we're in the nucleus, and sometimes it's hard to remember that there is other things in the cell, because all of this lesson is dealing with the chromosomes, which are in the nucleus. So we're simply talking about what is happening in the nucleus, what are the chromosomes doing, and then the rest of the cells divides accordingly. All right, this lady, they have her in here, and she gets very complicated after a while. So you can look at her on your own. I think the video I just showed you is a slightly more understandable than her because she just gets kind of carried away. So I'm gonna let you look at that one on your own. So again, what you need to know here is cell reproduction is called cell division. Okay. So we're going to do a little quick recap by looking at what this one has to say, and then we'll get into mitosis itself.
Hi there. Let's discuss cell reproduction. Before we start, let's begin by reviewing DNA. DNA stands for deoxyribonucleic acid. It's a type of nucleic acid, which is one of the 4 macromolecule groups. When a eukaryotic cell is not reproducing, its DNA is inside the nucleus.
All right. I'm learning that all of these things are AI generated, and they don't always phonetically pronounce everything the way it's normally pronounced. So again, deoxyribonucleic acid is DNA. This word up here is eukaryotic, and that simply means cells that have a nucleus, as opposed to, like we saw in the video, prokaryotic cells. They don't have a membrane-bound organelle called a nucleus, but they do have genetic material. So they have chromosomes, but it's just not encased in an envelope called a nucleus. So they also, so as the video said, they undergo cell division, but they call that binary fission. So that is a little bit different to what we're going to do with mitosis in eukaryotic cells. So most of what we're going to do is in the eukaryotic cell. Both our mitosis and meiosis that we talk about will be in eukaryotic cells. So again, remember, that is a cell that has an actual nucleus and the chromosomes are in the nucleus. Okay? Vocabulary.
As disorganized long strands called chromatin. Before a cell divides, the chromatin thickens and shortens into distinctly visible bodies called chromosomes. Each chromosome is a single DNA molecule. In order for a cell to divide, its chromosomes must first make a copy of themselves. This is DNA replication, so that each new daughter cell gets the original amount of chromosomes. Just after chromosome replication occurs, the 2 chromosomes, old one and new one, are stuck together. These 2 structures are referred to as sister chromatids. They are joined together by a central protein bundle called centromere. The sister chromatids are exact copies of each other, and each one will be distributed to one of the new daughter cells. Now that we have reviewed DNA, let's discuss mitosis. Cell division is the production of creating new cells. Cells divide in order to grow or repair damaged cells. An example is your skin or intestinal lining, which divide regularly to grow in size and or repair tissues. Not all cells divide though. A number of cells in your body, such as nerve, red blood, and muscle cells, do not divide once they are mature. The cell cycle is a timeline of events that occur during the life of a cell. The cell cycle is composed of mitosis and cytokinesis. The cell spends most of its time in interphase, which is the green area. During interphase, the cell is getting ready to divide. Interphase is divided into three sections, G1, S, and G2. In G1, the proteins are produced for the cell that will be necessary for cell division. In S, DNA makes a copy of itself, which is the DNA replication. In G2, cells make copies of the organelles. So mitosis is a type of cell reproduction that produces identical cells. This means producing cells that have the same DNA.
Okay, that's going to be a question you're going to be asked all the time. What is the difference between mitosis and meiosis? And the answer is that in mitosis, you make cells that are exactly alike, or at least that is the plan, to make exact copies and keep producing more cells of the same kind. When we get to meiosis, that is not going to be the case. Okay, all right. So here we go. So the first thing, actually, I want to, is it on here? Okay, I want to go back to there. Okay, you do need to know what the cell cycle is. That's first step number one. What is the cell cycle? And it is a process by which the cell undergoes changes. All the cells are doing this all the time. Every 24 hours, cells are going through this cell cycle. And as he said in the video, they spend more time in interphase than any other phase. And then this little orange section up here is the part that we're going to spend most of our time in. So we're not going to, so while the green part takes up most of its time, most of our lesson is going to be this little orange part over here. You will have to know what PMAT is. So that is going to be our first step, is to look at these things right here. So if you're taking notes, this is where you need to take notes, because you need to know what PMAT. So start by writing PMAT. Again, you should be taking notes. Start by writing PMAT. And PMAT is what you call the acronym that puts all of the phases of mitosis in order from where it starts to where it finishes. The other thing you need to remember is cytokinesis is not part of mitosis. Cytokinesis happens after. So mitosis basically describes what is happening in the nucleus with the chromosomes. Okay, so our first step, the P stands for prophase. So you want to look at, you have to know, because what you're going to get is a picture, and you're going to be able to look, you're going to have to look at the picture and say, what's going on in this picture? Is this picture about prophase? Is it about anaphase, metaphase? What's going on in this picture? And so your notes will help you look at the picture and say, in this picture, I see a nucleus with a clear membrane that begins to break down. So a lot of times the picture will show, instead of a full solid line, it'll show a dotted line going around in a circle. Okay, so the nucleus begins to break down, and the centrioles, which are the little structures on the ends, move to opposite sides of the cell, and spindle fibers help pull the chromosomes apart. Okay, so you want to write that down. Okay, so if you have it in your notes, you won't have to keep coming back to the page and Canvas in order to see it. All right, so that's prophase, that's the P. And then, as he was saying, things of you're thinking of another word to help you remember what's going on. The P is preparation. The cell is preparing the chromosomes, or preparing the nucleus to begin to divide. Okay, so prophase, preparing. So then, and I'm Gonna keep going because you can go in and click on these. I'm gonna give you time because I'm gonna stop talking long enough to do attendance. Um, in fact, let me do that now. You all go through, and I'll come back and go over these with you again. Go through and just click on each one and write down what they do. And again, he said metaphase, middle. He says anaphase. He said apart. Think of A for apart. And telophase, he said think of two cells, T for two. Okay, so write those down, and I'm going to stop sharing for a minute, do attendance, and I'll be right back. And hopefully you are writing these down, because again, you're going to get a page where you just have pictures, and you're going to have to identify what's going on in the picture. So make yourself a note. Which phase are you looking at? Okie dokie. Okay, I see some of you who are not doing anything at all. Everybody should be on the page for prophase. Okay, Keone, why is it I see you, but your page says that you're in algebra? Again, people, please use class time for classwork, and that way we don't fall behind. Again, it is very important not to try to be in two places as one, because you do have to pass this class. Uh, uh, uh. All right, Brandon, did you just get kicked out of your internet drop or something like that? Again, you all should be on camera. I got half of y'all who are not on camera. Folks, following rules is so important. The name of the lesson is, that's a good question. It's cell reproduction, and we're actually on the first part of that unit, mitosis, cell reproduction. Okay, Desiana, I saw your note. Cell reproduction, and we're on the part that says mitosis, and what you are doing is going through that box that says phases of mitosis, and you are clicking on the little plus sign, and you are making notes as to what it is going on during that phase. All right. Attendance. I got a lot of people who decided today one is not important. Sticky note. And Isaiah, I don't know what you're logged into, because it says you have no tabs open. So I don't know where you are. Okay. Yeah, no, I'm looking at when I look at Go Guardian, that's how I look at do the attendance. I look at Go Guardian, and so I'm seeing what you're doing, where you are, and what you're not doing. And sometimes a couple of y'all, it tells me that you've been idle for 30 minutes. Some of y'all been, because some of y'all log in just for attendance sake, and some of y'all are just doing something else. Everybody should be working on mitosis. Well, the fourth, what is PMAT? That's the question. What is PMAT? So next time I see you, you're going to have a picture, and your picture is going to ask you, what phase is this? So you are doing yourself a favor. Yeah, but that's not what I'm asking you. I'm asking you to write down what is happening during each phase. So when you see the picture, you can say, ah, this is prophase because the nucleus is starting to break down. This is metaphase because the chromosomes are in the middle. This is anaphase because the chromosomes are pulling apart. This is telophase because it's forming two new sets of nuclear material. That's what you're writing down right now. And you're writing it for you. You don't have to put it in the chat. This is for you so that you have something to look at rather than have to keep flipping back and forth to Canvas when you have to do the matching activity. And there will be one. And again, the videos with the young lady, she gets super complicated sometimes because she's so enthusiastic. So you're welcome to watch those. But I think the one that I have in the playlist might make a little more sense to you. So again, all you're expected to do right now is to, and I'll come back to share, is to write down what is happening in each phase. So again, we did prophase. And again, PMAT is your acronym for the phases in order, phases of mitosis in order. And you're going to have to remember it again for meiosis as well. But with meiosis, it gets a little more complicated because it's actually 2 phases on that one. All right. So again, folks, y'all are going to have to stay with it, keep up. Don't take a nap. Don't decide that you can multitask while you're doing this because this is a lot. I'm just telling you ahead of time, this is a lot. So you do yourself a favor and pay attention to everything the first time and keep up with your notes and actually look at them. We don't talk about homework all that much, but the only way to really do well with this is to actually go back after class and look at it again. All right. So, and that's just reading. That's not asking you to do anything other than to reread what you wrote. All right. So metaphase. Metaphase, again, the thing that you are to remember is it's in the middle. All the chromosomes are lining up in the middle. And spindle fibers form. So the spindle fibers are those lines that look like little hands that are coming, and they're going to attach to each one of those chromosomes. And they come from either side. So when we looked at a cell before, if we just looked at a whole cell, those little spindle fibers are usually hanging out together. And what they do during meiosis is they separate and they go to what we call the north and south side of the cell. So they go to the poles, okay? And as they go to the poles, these little spindle fibers start to form, these little fingers start extending, and eventually by the time we get to mitosis, they're going to attach to the chromosomes which are lined up in the middle of the cell. And they attach to the little thing in the middle called the centromere. Okay, so there's a couple of things you got to remember. Vocabulary, vocabulary, vocabulary. You got to know what a centromere is. You got to know what spindle fibers are. You know that each copy, so the chromosomes have already made a copy of themselves, and the copies are called sister chromatids. They're not sister chromosomes, sister chromatids. Okay, so know the difference between a chromosome and a chromatid. All right. Anaphase is our next one. This one is where, again, you remember it by the word apart. The spindle fibers start to shrink. And while they shrink, they are pulling those copies apart. So the chromosomes had made copies of themselves. They were held together at the centromere. Now they're going to pull those copies apart. Okay. And then telophase, the last one, you're going to end up with 2 new nuclei. Okay, 2. So the T for telophase is 2, 2. And right now, we're still not 2 cells. We're still just 2 nuclei in one bubble. Okay, but each of these new nuclei has its own set of chromosomes, the exact same number that we started with, because remember we made a copy. Okay, so each one of these has a copy, and that ends mitosis, because mitosis is just about what's going on in the nucleus. Now the cell is going to divide. Now that we have the instructions separated and created, then the cell and all the cell parts can divide into 2. So cytokinesis is just meaning cell divides in 2. It splits, the cell splits in 2, cytokinesis. So each one of those new nuclei is going to go in each part, and the organelles are going to split up as well and go to each cell. So each new cell has exactly what the first cell had. Okay? All righty. So what I want you to do with the time that's left is to go ahead and actually, let me go ahead and play this one for you. Make sure you wrote all that stuff down. What did that just do? Okay, hold on. See if it's actually playing. All right. There's obviously no sound with this. So you see, okay, let me pause it for a minute. All right, so this is what cells look like when they start. You see those little rod-shaped structures? Those are your centrioles. Those are the things that are going to move to each side of the cell when mitosis starts. And instead of being set together, they're going to go to what we call the opposite sides of the cell, or the poles. And we went through this last semester where the DNA replicated itself. So all the little nucleotides will line up, complementary nucleotides. They make copies. So it unzips, it makes a complementary copy of itself. I'm going to have to do the talking for it. And then it will zip back up, and then we can separate now the two sets of chromosomes. All right, so this is starting with prophase. And next class, we're going to actually go to a site where you actually can see this kind of happening in real time. But if instead of it being long, looking like spaghetti, because most of the time when you look at the cell under a microscope, it looks, all you see is this dense mixture of stuff. You can't really see chromosome. But once it starts to divide, then chromosomes condense, and you can actually see these little X-like structures. And the spindle fiber, technical name for spindle fibers are microtubules. And they reach out, and they're created from the centrioles. All right. Now we're all the way up to anaphase. I think I talked through metaphase. And it pulls them up to, like, again, we say opposite poles. So opposite sides of the cell, they're always referred to as the poles of the cell. In telophase, the nuclear membrane starts to form again around these new sets of chromosomes. And the spindle fibers disappear. It's not so much that they disappear, but they shrink back into the centrioles. Now, we're going to look at plant cells later on, because plant cells at this phase do it a little bit differently. So right now what we're looking at are animal cells, but plant cells are also eukaryotic, that they do have a nucleus. So they do everything up to cytokinesis will be the same in a plant cell. And like he said, we call them daughter cells. They are not son cells. So the two new cells are called daughter cells. All right, I'm gonna let this go ahead and stop, because we'll review this later on. Again, so again, here is another picture of everything you just saw in the video. So what we're going to do, we're going to look at a site called Cells Alive. So these pictures down here at the bottom are the kind of pictures you're going to see at Cells Alive. And you'll watch what they're going through step by step. And then, like I said, I'll have a worksheet for you to do that will ask you to fill in what's going on in this picture, what's going on in that picture. So hopefully you wrote them all down. Let me see what happens when you click on that. Oops, I didn't mean to stop sharing. All right, it forms a document that you weren't going to be able to see that. So it'll download a document for you. Okay. All right, so if you finish with all of that, give yourself a little quiz and do the drag and drop as a test for yourself. They've added pro-metaphase on here, but that's just the thing that's happening between prophase and metaphase. All right, I'm just doing this, but you should, but you'll be able to do this because you are looking at all of the information that is in these boxes up here. They don't have a pro. Yes, they do. All right, I'm going to put this on. They didn't like that. All right, so just keep. You see, and it won't let you put it in the wrong place. It'll jump back if you try to put it in the wrong box. All right. So again, you need to play with that. Make sure that you've quizzed yourself because you will have a quiz on this. And then go back and read the before you go. Know before you go. All right. If you have done all that you need to do, time is up. And I'll see you on Friday. Friday, right? Yep. No. See you on Thursday. Thursday. I don't know why I said Friday. Thursday. This week is there's no Wednesday class, so I'll see you Thursday. Thursday, yes, Thursday. All righty. So if you have written down all you need to write down, you are free to log out. I'm going to log out a little early because I do have another class immediately after this. Now I have a new class this